Hi everyone. This is the fourth lecture for your chapter Biomolecules. In the last class, we studied amino acids and proteins. So, till now, we have discussed all of the major classes of biomolecules. We started with carbohydrates, followed by nucleic acid, lipid, and then proteins. Today, we will be talking about secondary metabolites, enzymes, their structures, their types, their catalysis properties, and finally we will be talking basics of metabolism, okay, and the living state. Let us begin with secondary metabolites. We have already seen amino acids and proteins last time, fine. So what are these secondary metabolites? Secondary metabolites are defined as small organic molecules, okay, they are also small organic molecules which are produced by an organism, but they are not essential for their growth. Okay, so they are produced by the organism, but they are not required, absolutely necessary for the growth. Okay, they are just produced for some different function, but they are not involved in growth, development, or reproduction. If any molecule or any organic compound is involved in growth, development, and reproduction, it is called primary metabolite. Primary metabolite will include all the sugars, it will include the nucleic acids, it will include lipids, it will include proteins because these are all required for growth, development, and reproduction. While these secondary metabolites are not required. Okay. When we analyze plant, fungus, and microbial cells, okay, mostly you see secondary metabolites in huge quantities in case of plants, in case of fungus, and in case of different microbes, okay, different bacteria. Thousands of such compounds are found. Their roles and function in physiological processes is not yet understood. So, what is their function? Why are they required? Why is the plant or the organism making these molecules for itself? A lot of their functions are not yet discovered. Okay, some we know, some we do not know. Example is alkaloids, flavonoids, rubber. Okay, if you want me to give you example, alkaloids are just hydrocarbon compounds and you find them in different variety of plants. I'll give you an example of vinca alkaloids. Have you seen periwinkle? Periwinkle is called barmasi in Gujarati. So those barmasi flowers or periwinkle flowers, they produce some alkaloids, okay, some such sort of secondary metabolites. These vinca alkaloids are actually used for cancer treatment. Okay, so they have no such function in the plant itself, but they are very much useful for us. Then flavonoids are present in a lot of vegetables, and if you have ever seen or ever drank green tea, okay, green tea or black tea, all of these tea, they have a lot of color compounds. Okay, these compounds are the ones which give it the green or the brownish color, so they are flavonoids. Then rubber or latex is also a secondary metabolite. It is not required for growth, development, or reproduction, but still the plant is producing it. Then essential oils, see how the sandalwood, chandan, okay, it has a specific smell. Then lavender has a specific smell, rose has a very sweet smell, jasmine has a smell, right? So all of these smell, okay, they are called essential oils. So you can also have these essential oils which come under secondary metabolites. They are not required for growth, development, or reproduction. Antibiotics produced by fungus. Have you ever heard of the story of Alexander Fleming? Right? Alexander Fleming had a petri plate. Okay, on this petri plate there was penicillium growing. Penicillium notatum was the name of the fungus. This fungus did not allow bacteria to grow around it, and from there the discovery of antibiotics was made. So antibiotics are also such secondary metabolites released by funguses mostly. Then colored pigments, okay, like red colored beet is because of anthocyanin. Anthocyanin is a secondary metabolite. Same thing with purple cabbage or purple cauliflower. Okay, all these purple and reddish colored pigments are nothing but secondary metabolites. Then scent, as I had already told you, along with essential oils, such scents, then gum, Gundar Pak, have you ever heard of? Right? So you have gum which you use for eating, right? And then there are resins. Have you heard of amber? Amber, I think in a Gujarati માં પણ amber જ કહેવાય. So this amber, gum, they are nothing but they are secondary metabolites that you get from plants or trees. Ask your mother. એને પૂછજો કે ગુંદર છે એ normally અડદિયાની હવે season આવી ને, so in that અડદિયા also, in that traditional sweet also, you will find such uh, gum, okay, which is gundar, and that is produced as secondary metabolite. Then spices, why the peppers are hot, okay, આપણે તીખા જે છે એ કેમ તીખા લાગે છે. Then you have what you can say, cinnamon. Cinnamon is dalchini. Dalchini has its own taste and flavor. Then star anise, badia has its own flavor. Elaichi has its own flavor. So all of these spices have flavors because of secondary metabolites. So I hope you are getting the point. They are not essential for growth, development, or reproduction, but they are still produced by the plants or bacteria or fungus. Clear? Here is the what is the table from your textbook. Pigments can be carotenoids. Carotenoids are the orange colored pigments, okay, orange to yellow colored pigments that you find in the plants. From carotenoids, you get vitamin A. One molecule of carotene can make 2 molecules of vitamin A, so they are very, very important. That is why people tell you to eat more carrots, right? The carrot comes from this carotene because it is orange in color. Anthocyanin, as I already told you, seen in purple cabbage or seen in the beetroots, right? So this anthocyanin is also a pigment. Then alkaloids like morphine, codeine, etc. Morphine is given as ultimate painkiller when somebody cannot be recovered, okay, there is no recovery, there cannot be a surgery. Such soldiers are normally given morphine so they can die without pain. So morphine is an alkaloid. Then codeine, in previous years, okay, not previous years, but what can say, in olden times, codeine was given in cough syrup, okay, and this codeine, it is also an alkaloid. Then terpenoids. Terpenoids are just a type of secondary metabolite. It includes monoterpene, diterpene, etc. Remember this diterpene because gibberellic acid, okay, which is a plant growth regulator, is actually a diterpene. Clear? So remember this name, okay, diterpene or terpenoids. Then you have essential oils like lemongrass oil, and then you have even other spices and all which can make such kind of oils, okay, lavender oil, jasmine oil, rose oil. So these are nothing but essential oils. Some toxins meaning some poisonous compounds are also secondary metabolites. Example is heparin and ricin. Ricin is found in your castor seed. Castor is I think you ask your mother. Castor oil is put in your wheat, okay. Ghauma, castor oil in Gujarati શું થાય? It is called erandiu, okay. So that erandiu or that castor seed, it has this compound called ricin. In olden times, in Mughal empire, they used to give ricin as poison. Remember, if you are a spy, okay, you must have seen this in movies, if you are a spy and you get caught, then those spy, they eat something, some tablet or something, and then they die. So those kind of tablets are mostly either ricin or they can be cyanides, right? So ricin is one such type of toxin. It will block your transcription, okay, or mRNA will not be made. If mRNA is not made, protein will not be made. If protein is not made, then the function will be affected. So ricin is such kind of a poison. Then you have lectins like concavalin A. Concavalin A actually is nothing but a protein, okay, but it is a secondary metabolite. It does not have direct role in the organism's growth and development. Then drugs like vinblastin, this is the same thing which I told you, vincristin, vinblastin, you get from the periwinkle. Okay, so they are anti-cancer drugs. Then curcumin. Curcumin is this compound that you get from turmeric. Okay, turmeric, you get the yellowish colored compound is curcumin. Fine. Then polymeric substances, which are polymers. You have rubber, which is nothing but latex, gums, and cellulose. Cellulose, again, it is not required directly in growth, development, and reproduction, but it is required for structural support. Okay, so it is again a secondary metabolite. Clear? So here we end the secondary metabolites and we come back to enzymes. Enzymes are nothing but they are a type of protein, okay, functional proteins. So before talking about enzymes, let us just go back and discuss a bit of chemistry. Chemical reactions can be of different types. For example, if you take out ice from your refrigerator, you put it on a saucer or on a pan, and you see that it turns into water. If you heat this water, it turns into vapor. So this is also a chemical reaction of a sort. But what kind of? It is physical process. That is, no bonds are broken or no new bonds are formed. But there is still some change that you are seeing. The change is in the physical property, okay, from ice to water, water to vapor. Then there are chemical reactions where different bonds are made, different bonds are broken. For example, this is barium hydroxide, BaOH whole twice. Okay, barium because it is +2 charged cation. Okay, so barium hydroxide plus H2SO4, sulfuric acid. It gives BaSO4. So this SO4 is 2 minus, this ion, negative ion. So SO4 2 minus will react with barium 2 plus, give you BaSO4 plus H2O. So this whole thing is a chemical reaction. So reaction can also be of two types based on the energy. Okay, it can be exothermic or spontaneous reaction. Exothermic are the reactions which give out heat. Okay, when you see this reaction happening, it will give out heat. Okay, so they are also spontaneous. Spontaneous meaning they can occur on their own, they can happen on their own. And the second type of reactions are endothermic. Endothermic reactions, as you can see here, melting of ice, let us say. So this melting of ice requires heat. If you take one ice block and go to space where there is no there is vacuum, right? So in that case, that ice cube will remain ice cube, but only when it gets heat it will start to melt, right? So this kind of reaction is called endothermic. It takes heat inside. Okay, heat is required for its reaction, and this is energy requiring process. Burning of, let us say, wood, okay, it will be exothermic reaction because it gives out heat, and it is also spontaneous. Once you start a fire, it will keep on producing heat, right? Now when we talk about chemical reactions, we also talk about its kinetics. Okay, so rate of reaction, rate is nothing but anything upon time. Clear? So here what we are seeing the rate of reaction, which means the amount of products formed per unit time. So you have this amount of product formed, P, capital P, divided by the time. Clear? And this here will give you the rate of reaction. You can also write the rate of reaction as the amount of reactants used per unit time. But this definition is more used. Okay, the amount of product formed per unit time is more used, or more general form of the rate of reaction. The rate can be called velocity if direction of reaction is also specified. If you know the direction of the reaction, whether it is going forward or it is going reverse, you can call this rate of reaction as velocity. The rate of reaction becomes, okay, this is what I was going to say, yeah, in photosynthesis also we will be studying this, so pay attention. The rate of reaction becomes double. Okay, so let us say before you were eating 20 pani puris. in 10 minutes. Now the rate will become double means what? In the same amount of time you are eating 40 pani puris. Clear? So here the rate has increased, right? So the rate of reaction doubles when there is increase in temperature by 10 degrees. Okay, so let us say you were eating at 20 degrees Celsius, okay, in very cold condition, in a room which had AC at 20 degrees, and you were eating pani puri, 20 pani puris per uh, for 10 minutes. If you are taken outside where the temperature is normal, it is 30 degrees, then you are able to eat 40 pani puris, right? So the reaction rate doubles with increase in temperature by 10 degrees. Of course, there is a range. This will not keep on happening forever. Only from 20 degrees to 40 degrees, when there is rise in temperature by 10 degree, you will find this sort of relation. Okay, the rate of reaction becomes half with decrease in temperature by 10 degrees. Catalyzed reaction proceed at rates vastly higher than that of uncatalyzed ones. So catalyst, I hope you must have heard of in chemistry, right? So catalyst is nothing but it is a compound or a molecule which will increase the rate of reaction but not participate in the reaction itself, right? So such a reaction are called catalyzed reaction, and they are vastly they proceed at higher rates, okay, than normal reaction. Enzymes are nothing but they are catalysts in the living system. Clear? Now let us see the difference between inorganic catalyst, the normal catalyst that you use in chemistry, versus the catalyst present inside your bodies, which are enzymes. Inorganic catalysts, they work efficiently only at higher temperatures and higher pressures. Okay, they're very snobby. They require high temperature, high pressure, and some other higher parameters as well. Okay, here we'll just consider temperature and pressure. Example will be Haber's process. Have you heard of Haber's process? Okay, it is just making ammonia NH3 from nitrogen and hydrogen. So nitrogen and hydrogen, they react in presence of catalyst, which is nickel or iron, and you get ammonia. This process happens at 400 to 500 degrees Celsius temperature and 150 to 250 atmospheric pressure. Yeah, so can you see this catalyst will only be able to work when there is such high pressure and temperature. Enzymes, on the other hand, they work efficiently at body temperature. Okay, they are very happy at 37 degrees Celsius. If you take it below 37 degrees, their function will be affected. If you take it above 37 degrees, their function will be affected. They denature above 40 degrees Celsius. So if you take them above 40 degrees, they lose their shape, they lose their function, and that is called denaturation. We shall see this later on. Exception to this number are the enzymes which are isolated from thermophilic bacteria. Again, try to remember biotechnology. You must have heard of Taq polymerase in PCR. Taq polymerase is isolated from your Thermus aquaticus, which loves to live in high temperature water. Okay, so these enzymes are isolated from 80 to 90 degrees Celsius environments, and that is why they are stable at such higher temperatures. But normally, other enzymes are all working efficiently at body temperature and they lose their function above 40 degrees. Example of the same process of making ammonia in case of biology. Okay, in biological process, if you see nitrogen fixation, this occurs at normal pressure and normal temperature. It does not require 500 600 degrees. It does not even require 150 250 atmospheric pressure. It happens at normal temperature and pressure. Yeah, so this is the difference between inorganic catalyst and an enzyme which is organic catalyst. Now let us talk about enzymes. There are two types of enzymes. Always remember this. Okay, it is very, very important. One type of enzymes are made up of RNA. Here what you are seeing is this single strand of RNA which has formed this sort of 2D or 3D structure. Okay, it can fold on itself and form this kind of secondary structure. RNA. Such kind of enzymes are called ribozymes. So all enzymes are proteins except the exception is ribozymes, which are made up of RNA, as you can see here. Okay, so some RNA can also act as enzymes. Example is peptidyl transferase that you see in your ribosomes. Okay, remember when we were discussing organelles, we discussed ribosome and I told you it has an enzyme which is made up of RNA, and that enzyme is called peptidyl transferase. We will also discuss this when we talk about translation, okay, molecular basis of inheritance, and at that time also we will see that this enzyme is actually made up of RNA. All the other enzymes are proteins and they are tertiary structure with lot of folds and crevices. You can see a lot of folds, you can see a lot of crevices. Okay, crevices are nothing but they are slits, okay, or depressions. So can you see here there are a lot of ups and downs and the structure is such. This is the 3D structure of an enzyme. Clear? And it has a pocket. Fine, it has a cave-like structure. This pocket that you see here is nothing but the active site. Okay, one such pocket where the substrate binds is called active site. Substrate is nothing but it is a reactant. Okay, in the reaction when it binds, it is that place is called active site. They greatly increase the rate of reaction, and let us see the whole process now. Okay, one more thing. This active site is very specific. It will allow only one type of molecules to bind to it. Okay, as we shall see in this example. So this is the active site. These are the 3 types of substrate, and you can see that this substrate cannot bind. Okay, then they will try with another shape of substrate. So it will try to bind this one type of substrate, but it will not be able to bind. Then this will also try to bind, but it will not happen. Okay, and then finally its original substrate which will bind to it like lock and key. Okay, so this is called lock and key model, will get fixed, and then this enzyme will be able to do its function. As you can see here as well, this is the enzyme. It has the specific active site, and then here it is actually sucrose. Okay, the enzyme here is sucrase or isomaltase. As you can see here, this is the sucrose molecule, glucose plus fructose, and one molecule of water has come, and it can only get attached here in the active site. Only sucrose can attach here in the active site of the enzyme. This is sucrose, that is water. It gets added and glucose and fructose are separated, right? So let us take an example of this reaction. Carbon dioxide plus water gives you carbonic acid H2CO3. This you must have studied during respiration, okay, or breathing and respiration, okay, that chapter when you discuss lungs and or you might have discussed in RBCs. Okay, so same thing here, you have carbon dioxide plus H2O, it gives you carbonic acid, and the catalyst for this reaction is carbonic anhydrase. Okay, carbonic anhydrase. And this reaction can go backwards as well. Carbonic acid can dissociate to form water and carbon dioxide, but this shorter arrow shows that it is slower than this forward reaction. Fine. So without enzyme, carbon dioxide plus water gives you carbonic acid. This whole reaction, it forms only 200 carbonic acid molecules per hour. So in 1 hour, you get only 200 H2CO3 molecules. With enzyme, ready to be amazed, with enzyme carbonic anhydrase, you get 600,000 H2CO3 molecules per second. Okay, just calculate in hours how many you will get in hours. Okay, so per second you are getting 6 lakh H2CO3 molecules. So this is how much the enzymes speed up the reaction. The enzyme has accelerated the reaction rate by about 10 million times. Fine. Now let us see how the enzymes actually function. Like, how are they doing this? So the substrate, it binds to active site and forms enzyme substrate complex. It is written as ES. Fine. So E plus S gives you ES. This is ES. Okay. This is the active site. This here is your substrate, the rod shaped substrate, and this blue colored one is your enzyme. This is your ES complex. Now what happens is, after the substrate binds, a new structure of substrate called transition state is seen. So just think, if it was exactly lock and key, how would the enzyme be able to work? Instead, it is somewhat like this. When the substrate binds, this enzyme can function, okay? It can do some work. How will it do this work? So it will, just think of, let us say, a chalk in between your hands. And if you try to push it, it is bending. Okay, it will bend to fit this active site. So this is called transition state. Okay, before breaking, it is bending a little. So this is ET, enzyme transition state complex. After this, the enzyme will break the transition state and give you the two products. Okay, so this is E plus P, here the enzyme and the products. Clear? So this is just an illustration of how the enzyme actually works. The substrate, it binds to active site, then the substrate undergoes some change. It will be under some pressure, it will be under some pulling and pushing, and then only that stage is called transition stage, and after that transition state only there will be synthesis of products. The enzyme catalyzes bond breaking or making, and the product is released. In this case, the bond was broken between this substrate. There could be many more such altered structural state. There could be many such transition state before finally the product are formed. Right. So let us see the graph for this. Just pay attention, okay? On the y-axis, you have potential energy. Potential energy is the energy present in the molecule, right? And on the on the x-axis, you have progression of the reaction. In which direction the reaction is progressing. So the substrate, it normally has a higher energy compared to the product. Okay, you can only make a product which has lower energy if the substrate has higher energy, right? So you start with substrate which normally has higher potential energy, and you end with the product which has lower potential energy. Clear? Now what is done to these substrates? These substrates, when they are transformed into transition states, okay, as we saw that the substrate was bent. So this bending, it requires a lot of energy, okay, which is why the transition state level will have higher energy compared to the substrate, because we have done a lot of work on it. Okay, you have tried to push that chalk, and so you have done a lot of work on it. Now that chalk will have a lot of energy before it breaks. Clear? So just pay attention here first. So you start with the substrate. You are taking the enzyme is working on it, so it is moving up to transition state, and this transition state has higher energy than the substrate. Once this transition state is there is making and breaking of bonds. Once that has happened, it will break down into products, and these products will have lower potential energy. The difference in average energy content of substrate and the transition state is called activation energy. This is the energy you have to give for the substrate to turn into products. Okay, so the difference of energy between transition state and the substrate, okay, is called the activation energy. So here, this is the activation energy without enzyme. And the second pink colored graph that you are seeing is the activation energy with enzyme. Can you see the difference? This means that when enzymes are present, the transition state can be achieved with help of less amount of energy. Okay, so it brings down the activation energy. That is what enzyme does. And because this activation energy is brought down, now a lot of substrate can be converted into product with less amount of energy. Clear? That is what is seen in this whole graph. I hope you are able to understand. To understand it better, let us think of this analogy. There is this barrier. Okay, let us say there is this wall, okay, made up of stones, and there are these many frogs. These are nothing but reactants, okay, which are at higher energy level, and these are the products which are at lower energy level. Now the frogs have to jump across this wall and then reach this level. Okay, but as you can see, only few frogs will be able to jump. Not all frogs will be able to cross this barrier. Now let us say that you broke down this wall, you took out some of these stones so that the wall is smaller. Now a lot of frogs will be able to go from this side to the other side, from reactant side to the product side. This thing is done by the enzymes. It is just removing this barrier. Okay, it is removing the activation energy barrier so that more reactants can go into the product uh, side. Okay, fine. So that was all about enzyme, its structure, its catalysis, how it is doing, and finally the graph, okay, which shows that it brings down the activation energy, and so the reaction can proceed forward. One more thing I wanted to tell you is enzymes will only be able to perform reactions which are spontaneous, okay, which can happen on their own. They will not be able to do any impossible reactions, okay. They are just doing the possible reactions but at a faster rate. Clear? Okay. Now let us talk about factors that affect its enzyme activity. Okay, the enzyme activity is affected by number one, temperature and pH. Now the enzymes are present inside our cells, inside our body. So they function in a very narrow range of temperature and pH. They like some temperatures and some pHs only. Okay. Such enzyme show its highest activity at a particular temperature and particular pH. That particular temperature is called optimum temperature and that particular pH is called optimum pH. When it is doing its best work, it is called optimum temperature or optimum pH. As you can see here, this is the diagram. Here you have activity of the enzyme and this is here the temperature. As the temperature from 0, let us say, it keeps on increasing, you can see the enzyme activity is also increasing. It will keep on increasing, it will be its best. And then let us say after 37 degrees, 40 degrees, after 40 degrees, the enzyme activity will keep on decreasing. What is this bloody arrow? Okay. This after, as the temperature keeps on increasing, the activity keeps on decreasing. So it will keep on going down. Clear? At lower temperatures, the enzyme is an inactive form. Okay, at lower temperature, the enzyme is just frozen. It cannot act. While at higher temperature, it denatures. It loses its 3D structure, which is called denaturation. So it denatures, and that is why you see decrease in the activity. Same thing happens with pH. Okay, this is denaturation. So this is your normal functional protein. And when it denatures, it opens up like this. Okay, it becomes unfolded. And this unfolded protein cannot function properly. Okay, this unfolded enzyme cannot function properly. And that is why you see that reduction. Okay, here. Same thing is seen with pH. At lower pH, the enzyme will not work. It will not work. With increase in pH, it will keep on increasing the rate of reaction. The enzyme activity will be seen. At a specific pH, for our body, let us say at 7 pH, you will see that the enzymes are working best. This graph or this optimum pH will be different for different enzymes. Think of your stomach enzymes. Okay, pepsin works at 2 pH or less than 2 pH because your stomach is acidic in nature. Okay, so the pepsin enzyme, it works at its pH optimum is 2. Same way, if there is some cytosolic enzyme, its pH optimum will be 7. So this enzyme is working best at the 7, then it is called its optimum pH. And if you increase or decrease that pH, the activity of that enzyme will also decrease. Fine. Second factor affecting enzyme activity is concentration of substrate. How much substrate you are giving to the enzyme. At first, the substrate concentration is proportional to the velocity of reaction. Just look at this diagram. You have velocity on y-axis. Velocity is nothing but rate of reaction. But when you know that it is moving in forward direction, that is, you know the direction of reaction. So you can call it velocity. So this is velocity of the reaction. And on the x-axis, you have the substrate concentration. At lower substrate concentration, as the substrate concentration, it keeps on increasing a little bit, the velocity also keeps on increasing. So you can see this linear rise, okay, which is proportionality. Okay, that as the substrate concentration is increasing, so the velocity is also increasing. So you can see it is proportional. After some time, it reaches its maximum velocity. Maximum velocity is that the highest rate it can reach up to. Okay, now just look at this diagram again. At the beginning, these blue colored C-shaped molecules are nothing but your enzymes, and the red colored ones are your substrate. So at the beginning, the substrate concentration is comparatively lower than the enzyme. So more and more substrate gets added, more and more reaction will move forward. So you can see more products being formed, and this is the rate of reaction. It will keep on increasing. So this is the point B, where there are comparatively more amount of substrate, as you can see here, for the enzyme to work on. Then at this point C, you can see that all the enzymes are already working on the substrate. So there is a little extra substrate remaining in the solution. Fine. So this means all the enzymes are working at their best possible capacity. After this point also, if you keep on adding more and more substrate, then the enzymes, they will be able to work only at their best rate. Okay, they will not, let us say if you can write 100 letters per minute, then you will only be able to write 100 letters per minute. Does not matter whether you have to write a 3-page essay or a 30-page essay. Same way here, the enzymes, they can only do reactions in their proper time, okay, at their own rate. So now the substrate level is higher, but the enzymes are still working at their maximum capacity. So the this graph, it will become straight line. Okay, now even if you keep on adding the substrate, your enzymes are not free. Okay, they are already busy converting the substrate into product. So they will not be able to convert this added substrate into product. Okay, and so you will see no rise in the rate of reaction. This is called its maximum velocity. Okay, here all the enzymes are working at their best potential, and you will find the maximum rate or maximum velocity. It is known as V max. After this, increase in substrate concentration has no effect, as there are no free enzyme molecules left for additional substrate molecules. Clear? Just understanding this graph a little, because once or twice the questions were asked on this. So this is the Vmax. Okay, normally you leave a little bit of gap because this line will continue straight. So you are just writing it like this. Okay, the Vmax, a little bit above. So this is the Vmax, the maximum possible velocity. Here you have substrate concentration on x-axis, reaction velocity on the y-axis. The half of Vmax, that is, the reaction will be at half of its maximum speed when the concentration of substrate is Km. Okay, so this Km is nothing but the concentration of substrate. Okay, Km is concentration of substrate when the velocity will be half of its maximum. Clear? This is the V max by 2. This is V max, then this is V max by 2. This V max by 2, wherever it meets on this graph, and you project it on the substrate concentration. This is the substrate concentration, Km, where the velocity is half of its maximum. Clear? This is at a lot of places, Vmax and this Km are used to analyze the enzymes. So it is important parameters. Other factors affecting enzyme activity are inhibitors. Okay, these are certain chemicals that do not allow enzyme activity. Okay, the enzyme to function properly. This process is called inhibition, and these molecules are called inhibitors. An example is you can have different types of competition. Just let us talk about you can have different types of inhibition. Let us talk about two. There is competitive inhibition. In competitive inhibition, the substrate and the inhibitor, they have similar kind of structure. Because they are similar in structure, they will fight for the active site. There will be competition for the active site. Think of let us say first rank, okay, and there is a 100-meter race. So there will be a competition between two participants. Same way here, there will be competition between the substrate and between the inhibitor to bind to the active site of the enzyme. So this is called competitive inhibition. If the inhibitor is in higher concentration, it will bind. Because this inhibitor is not original substrate, okay, the enzyme, it will bind to the enzyme, but the enzyme will not be able to do its function. So this is competitive inhibition. The non-competitive inhibition is when there is an extra site on the enzyme, which is called allosteric site. Okay, think of it as your scissor. Okay, let me just draw. Let us say you have this scissor, and if you fix something here, okay, you're not allowing this scissor to come these holes to come together, okay, you're not allowing this scissor to function properly, then will it be able to cut anything? Okay, let us say okay, here, let us say that I have fixed this position, so the scissor will not be able to cut anything, right? The same way this site will be known as the allosteric site. Clear? The arrow is better. Right. So this site is the allosteric site. If something comes and gets fixed here, the substrate will not be able to bind at the active site. So this is called non-competitive inhibition, because there is no competition between the two. They are not same in structure. But still, this another molecule, which is the inhibitor, if it comes and binds on the allosteric site, which is the other site, okay, if it binds to the allosteric site, the active site will no longer remain open, and so the substrate cannot enter. Example of such competitive inhibition is inhibition of succinate dehydrogenase. Remember, succinate dehydrogenase is an enzyme you find in the TCA cycle. So the succinate dehydrogenase, it normally binds to succinate, which is this molecule. It binds to the succinate, and then you get fumarate plus the enzyme. Here, this is the reaction. At some in some places, okay, what happens is instead of succinate, a similar compound called malonate can bind. Okay, here is malonate. As you can see, it is very much similar to the succinate, right? So this malonate, if it binds to the enzyme, it will not allow the enzyme to function. So there will be no product formed, but that enzyme will be inhibited because now succinate cannot come and bind to this place. Clear? So this kind of inhibition is competitive inhibition. In such a scenario, if you add a lot of succinate, then this malonate will be removed and succinate can come and the enzyme can function properly. So competitive inhibition is reversible. If you increase the amount of substrate in comparison to amount of inhibitor, then it can be reversed. The use is to control some bacterial pathogens. To control some bacterial pathogens, such kind of inhibitors are given as drugs, and so the bacterial enzymes will not be able to function properly and the bacteria will stop functioning at all. Okay, and this is how you can use it in day-to-day life. Now the final segment, which is classification and nomenclature of enzymes. Enzymes perform a variety of different functions, okay, variety of different reactions. They are classified in 6 major types. Again, remember, this series should not be changed, okay? These are numbered and that number should not change. The first class will always be oxidoreductase or dehydrogenase. Second is transferase, hydrolase, lyase, isomerase, and ligase. Let us see them one by one. Oxidoreductase type of enzymes or dehydrogenase type of enzymes perform the reaction of oxidation reduction, okay, or redox reaction. Example here, as you can see, this A or this blue colored molecule is reduced. It has these two hydrogen atoms, or let us say electrons, okay. It is reduced, and here the B is oxidized. What will happen is, let us say this A is transferring its electrons or hydrogens to the B, and this whole process is called oxidation reduction reaction, and this type of reaction is performed by oxidoreductases. Example is succinate dehydrogenase, which we just saw right now, okay, in competitive inhibition. Some enzymes are transferase type, which means they transfer a group from one compound to another. So you can see here, this is AB. The B is this yellow colored portion, and this C is another substrate. What the enzyme will do is, it will take this B, okay, this group from the first substrate and transfer it to the second substrate, which is C. It makes this DC type of molecule. Clear? So this transfer reaction from one substrate to another is carried out by transferase. An example is hexokinase. Remember, hexokinase is the first enzyme you find in glycolysis, and what it is doing, it is taking phosphate from ATP and transferring it to glucose. Clear? Third type of enzyme is hydrolase. Hydro means water, okay, and lase means to break, lysis. So it does the function of hydrolysis, which is breaking of a bond by addition of water. As you can see here, you have this AB compound and a water molecule. Water molecule will add, let us say, one hydrogen to A and one OH group to B, and this is how it will separate the two molecules, okay. It will give two products. Example could be amylase, which is breaking down your starch, or it is breaking down your glycogen into glucose. Clear? Then you have pepsin, which will break down polypeptide into peptides, or trypsin also, which does the same thing. It will break the peptide bond by addition of water. Okay, so these are the hydrolase type of enzymes. Remember, we discussed acid hydrolases, okay, which are present in the when we were discussing lysosomes. Clear? So acid hydrolases we discussed there. Then you have lyases. Lyases will break the bond without addition of water. Okay, so here you have A and B, and they will break that bond between AB, okay, without addition of water. Example is decarboxylases and aldolases, which we will again see during glycolysis. Okay, isomerase, which convert one isomer into another isomer. Example are epimerases and mutases. And finally, you have ligase. This type of enzyme, they make bonds, okay, using ATP. And the example is DNA ligase, which we will see in replication, okay, or we will also see it in your biotechnology chapter. Clear? So these are the 6 different types of enzymes, 6 different classes of enzymes. Any enzyme that you see will always be put under one of these, and this what you can say, the order is very much important. Okay, it will always be first oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. Fine. Enzymes, they require some cofactors to work. Okay, these are mostly non-protein type of compounds. Okay, and they make the enzyme catalytically active. So the protein part of an enzyme is called apoenzyme. Then along with cofactors which make it active, okay, which activate the protein, it makes it a holo enzyme. Just remember holo enzyme as whole. Okay, WHOLE, whole. So it is a complete enzyme only when apoenzyme or the protein part is along with the cofactors or the non-protein part. The cofactors can be of different types based on its connection or interaction with the enzyme. It can be metal ions. Okay, cofactors can be metal ions. Example is zinc for carboxypeptidase. It is an enzyme you will see in digestion. Okay, carboxypeptidase is an enzyme which breaks the amino acid from C terminal. Okay, then carbonic anhydrase, which we just saw, it catalyzes the reaction of carbon dioxide and water. So it has, as you can see here, can you see the zinc molecule, metal ion is present in the active site of the enzyme, and then you have this water molecule and carbon dioxide which will be converted into carbonic acid. Yeah. Then the cofactors can be prosthetic groups. Prosthetic groups are mostly organic. Okay, they are vitamin derivatives and they are tightly bound. They are covalently attached to the enzyme. Example is heme in catalase and peroxidase. Remember, this was the structure that I drew when we were discussing the hemoglobin, right? So this here is a porphyrin ring, and you can see the iron molecule inside. Okay, iron atom. Sorry, my mistake. Iron atom inside, and so this whole porphyrin plus the iron is called heme, and this kind of heme group is nothing but a cofactor, a prosthetic group which is found in catalase enzyme and peroxidase enzyme. Peroxidase, again, catalase and peroxidase we have heard about in peroxisomes. Yeah, what they do is you have hydrogen peroxide, so that hydrogen peroxide will be acted upon by catalase to give you water plus oxygen. And finally, the third type of cofactors are coenzymes. Okay, coenzymes are organic, again, just like prosthetic group, but they are in transient association with the enzyme, which means they are only for some time attached to the enzyme, and then they are removed once the reaction is complete. Example is NAD, okay, nicotinamide adenine dinucleotide, NADP, etc. for oxidoreductases. Yeah. So this is the table for different enzyme cofactors. You have TPP or thymine pyrophosphate for pyruvate dehydrogenase enzyme, okay, which is also another enzyme we will study in respiration. Then FAD enzyme as coenzyme, nicotinamide adenine dinucleotide, NAD, pyridoxal phosphate, coenzyme A. All of these coenzymes, or at least most of these, we will discuss in respiration chapter. Clear? And as you can see, thiamine, then flavin comes from riboflavin. All of these are vitamin derivatives. Fine. So there was a question once that coenzymes are mostly derivatives of vitamins. Clear? Metal ions could be zinc in case of carbonic anhydrase, carboxypeptidase, then magnesium in case of EcoR5. EcoR5 is a restriction endonuclease, which we will study in your biotechnology. Okay, and hexokinase, again, it has magnesium, nickel in case of urease, molybdenum in case of nitrate reductase, which we will see in nitrogen metabolism, biological nitrogen fixation. Clear? So different enzyme cofactors and coenzymes are required for enzyme activity. Okay. Let me just check the time. Fine, can complete. Chalo. A system at equilibrium cannot perform work. Okay, normally when a system is at equilibrium, Let us say the forward reaction is equal to the reverse reaction. In such cases, it cannot perform any work. Okay. Um, for in like just to visualize it, think about two water tanks at the same level. Will there be any flow of water? No, right? So at equilibrium, they cannot perform work. Living organism, they work continuously, like how I am speaking, you are listening, you are thinking, you are writing down notes. So living organism, they are working continuously, which means they must be not around equilibrium. Okay, their system should not be around equilibrium, so they cannot afford to reach equilibrium. The only time living organism reach equilibrium is when they die. Okay, the dead body is at equilibrium. Whatever is outside, the same is inside. Yeah. So living organism, they work continuously, so they cannot afford to reach equilibrium. Hence, living process, okay, the process of living, surviving, is a constant effort to prevent reaching equilibrium. Your body is constantly trying to not reach equilibrium. Fine. And this can only be done by input of energy. You have to apply, or you have to give a lot of energy just to make sure that you do not reach the equilibrium. Where does this energy come from? It comes from metabolism. Clear? Metabolism provides mechanism for production of energy. Hence, living state and metabolism are synonymous, because you cannot be alive unless there is metabolism going inside your body, which is producing energy. This energy will be used to stay away from equilibrium, okay, to be able to do work. And this whole thing is called the living state or the living process. So you can say that living system or living process and metabolism, they can be synonymous. Okay. Without metabolism, there cannot be a living state. Without metabolism, just think about your mitochondria. If your mitochondria is not working, ATP is not produced. If ATP is not produced, a lot of reactions in your body will stop, and eventually it will lead to death. Right? So if you are alive, it means there is energy being given to the system, and this energy is produced from metabolism. Clear? Just to show you this, the same water tank example I have shown here. If the water tanks are at same level, there will be no flow of energy, right? There will be no work done. But if one has higher level of water or is at higher level compared to the other water tank, there will be flow of energy. There will be work done. Clear? So living state is like this. There is always some work that is being done. You have concept of metabolism. Biomolecules, they have turnover. That is, if glucose was synthesized today in plants, it will not always remain glucose till the end of time. Okay, it will get converted into triose sugar. Then from trioses, we will make ATP. Okay, so it is always under turnover. Fine, they are constantly being changed into some other biomolecules, or they are also being made from some other biomolecules. Think of starch. Okay, from starch you are breaking it down and releasing glucose. So this way biomolecules are always getting converted into something else, and so they have a turnover. This breaking is called catabolism, and the making is called anabolism through chemical reactions. In total, okay, this breaking and making is known as metabolism. Okay, catabolism plus anabolism is metabolism. Did I make a mistake? Catabolism, anabolism, and metabolism. Okay. Then metabolic pathways, mostly whenever you talk about any reaction, it is hardly ever done in one step. Okay. Let us say conversion of glucose to ATP, it requires many steps. So all these steps and all those enzymes combined, they form a metabolic pathway. For example, from glucose to 2-pyruvic acid, which is nothing but your glycolysis. This glycolysis, okay, from C6H12O6 plus oxygen to give you C3H4O3, which is pyruvic acid plus water. So this whole reaction, it is not a single step reaction. It happens in multiple steps, in multiple reactions with help of different enzymes, 10 different enzymes. So it is called a metabolic pathway. Here, the glucose is substrate and pyruvate is the product. There are multiple pathways which can start from glucose and end in different products. Example, there is lactic acid fermentation. Okay, so you can start with glucose. This is just an example. You can start with any biomolecule. Here we are starting with glucose. You can get pyruvate also by 10 reactions of glycolysis. After pyruvate, this can also be converted into ethanol in case of alcohol fermentation, or lactate in case of lactic acid fermentation. If not, then by aerobic respiration, this pyruvate will be converted into carbon dioxide and water, and you will get a lot of ATP. Clear? This is called aerobic respiration. So the metabolic fate of glucose, what we discussed first was the metabolic pathway, and the whole thing is called metabolism. And here the biomolecules, or which as example we took of glucose, can have multiple fates. Fates is bhagya. Okay, it can have different bhagya. Right? So there we end the chapter biomolecules. Just solve as many questions as you can, and if you have any doubt, let us discuss those doubts. Clear? It is a very simple chapter, and I know you have already studied it twice in the school, and you must have studied it also when you were appearing for NEET. So I took it a little fast, okay, instead of slow, because it is a simple chapter, but again it has a lot of weightage in terms of marks. Okay, so solve a lot of questions, and if you have any doubts, let me know. And next time we will be starting with a new chapter, which is photosynthesis. The reason being, just when your biomolecules is fresh in the mind, right, you will be able to understand photosynthesis much better. Clear? See you next time.